enrIceTrait, a RiceTrait enrichment tool with ontology-guided text-mined evidences. We will introduce our project from 8 aspects, including the work we have finished and the job we are still working on. First, the background of our project. Our project aims at mining gene, trait, and gene-to-trait associations from the literature. As this figure shows, the mined genes are highlighted in orange while the traits in green. The orange line shows the co-occurrence association between gene and trait in a sentence. We then combine our manual results with 3 existing databases, and we ended up with a Python and R package to realize a function on the RiceTrait enrichment. Meanwhile, an integrated RiceTrait ontology, RTO, can also be made for public use. The reason that we mine this gene-to-trait association is that it has long been a focus for crop molecular breeding. In the preliminary research, we found that Gremlin was the first comparative genome database for cereal crops and model plant, and also the first to offer a trait ontology, TO. And we also discovered the first gene-to-trait database for rice, enrRiceTrait. In addition to enrRiceTrait, there are 2 other projects that have done similar work, TAS and FranceGene. The table clearly shows the information of the species, the quantity of associations, the data source, the method, and the time, etc. of the databases respectively. You can see that the TAS is manually curated, so it is time-consuming. However, FranceGene used an automatic way, and its result is highly recognized, but it's just based on a basic NLP method. Here are the details of the 3 existing databases. The quantity of gene-to-trait associations they provide was shown on the left. As you can imagine, there should be some crossovers between the gene-to-trait associations in these 3 databases. But figuring out the exact number of the crossovers is a difficult task because the source data they adopted are different, such as the normalized gene database they used. The proportion of the number of genes used in these 3 databases to the number of complete rice genes is shown on the right. In this project, we have to solve following NLP scientific problem. Though there are many challenges, such as the domain resources is backward and the research teams in this segment field is limited, etc., meanwhile, there are opportunities. As we mentioned on the last slide, concept normalization is one of the NLP scientific problems we encountered. We are faced with a situation where the normalization of gene concepts is relatively fixed and easy by using the existing rice gene normalization database, as the example shows, whereas the normalization of rice traits is faced with some technical difficulties. For example, in natural language texts, only a few mentions can be strictly matched to ontology concepts. Most of the trait mentions are not totally consistent with ontology concepts, for example, the total N and the term nitrogen content in TO. And some just have similar concepts like N uptake efficiency and nitrogen harvest index, while some, such as N distribution, have no corresponding concepts at all. Here is the workflow of our project. Integrate the data from existing databases and the data of gene-to-trait association that we have mined from the text, and normalize the data respectively. Then a Python or R package was made using the integrated results, and a RiceTrait ontology was produced at the same time. We collected the data from PubMed and the quantity of PubMed rice literatures from 2000 to 2021. As you can see, the amount of rice-related literatures is skyrocketing. In addition, we also do the literature classification according to the impact factor, etc. You can filter enrichment results by category of literature you want. Gene mention is the description of gene in the text. Lysine histidine type transporter 1 and also its abbreviation are the examples, and we highlighted them in orange. Gene symbol was obtained after rice gene annotation. The tool we used to do the gene annotation includes Genome Plus, Huner, Hofler. Once we get the gene symbol, we will do the dictionary mapping using enrRiceTrait gene dictionary, as shown on the right. We found the corresponding MSU ID and the RepDB ID for each gene symbol. This process is called normalization. The annotation resulted by different tools as shown in this table. We used Hofler in our project in the end because it had the highest F-score. RTO construction. First, we will introduce the cornerstone of our RTO, plant trait ontology. PTO is a controlled vocabulary to describe phenotypic traits in plants. The first few databases like Gramining and enrRiceTrait employ TO to facilitate trait-related phenotypic curations. It has 1,554 items, and each item includes the information like class ID, preferred label, synonyms, definitions, obsolete or not, and so on. Plant trait is an example. We further researched several existing ontologies with the crop to construct a more complementary RTO. Here we show the roadmap of our future work in building the RTO. Now we would like to introduce the detail of our first version of RTO. We have already known that the PTO included normalized plant trait conception, while WTO included the weight trait conception and environment condition. Also, WTO is the first crop trait ontology. Obviously, it is worthy of inclusion in our RTO. Next, we will introduce specific strategies of our ontology integration. We complete this work from 3 aspects. First, the merge strategy. If the WTO term has the same term as TO, then we map it to the corresponding TO term along with the original WTO hierarchical structure. We merge 75 nodes totally. Second, the add strategy. Add the term related to RiceTrait in WTO to TO and keep its WTO ID and child nodes. We successfully add 118 nodes to TO. Third, the delete strategy. If the WTO term is not related to RiceTrait, then we delete it. We totally delete 63 nodes. Now, the RTO 1.0 version has completed. It includes 2022 RiceTrait concepts now. You can download the RTO from this page or scan the QR code. In our future plan, the 526 keywords of fungus gene will be added to the RTO 2.0 version with the help of the domain experts. Some of the keywords are shown on the right. Currently, we need collaboration to construct a more complementary RTO to solve some of the problems that we face right now. We have completed the rule-based method, and the semantic computing and deep network-based method is ongoing. First, the rule-based method. We use the concepts in RTO as the seed matching words, but they are insufficient, so we expand these seed words from 2 aspects. One way is using the NLTK for part-of-speech reduction, as the below example shows. The other way is replacing the suffixes. For example, the root quality trait can be matched with root quality. Since this method is based on the trait concept, there is no need to do the normalization. We first extract non-phrases by using the NLTK toolkit. Then, based on the deep semantic networks like BERT, BioBERT, Word2Vec, etc., to encode the trait concepts respectively. And then, based on cosine similarity, we generate the candidates of rice phenotypes. Then, re-ranking the candidates by using the jacket index, and the first trait is the standard phrase we want. Rice gene-to-trait association extraction. For the rule-based method, the gene-to-trait association extraction is based on the co-occurrence at sentence level. Extract sentences containing at least 1 rice gene and 1 rice trait, as the figure shows. In order to decrease the false discovery rate, we adopted several methods as follows. And then we used Venn diagrams to show the gene-to-trait associations we extracted and also show the crossovers with existing databases. Our Python or R package for enrichment analysis. The first version provides with 4 sources: enrRiceTrait, TS, precise matching, and semantic computing for the rice gene-to-trait enrichment analysis. You can input the normalized ID of a set of genes to be queried, and set the parameters by selecting the source data from the 4 data we mentioned above. Then you will get the output of enrichment result of a set of rice genes on the concept of normalized trait. Acquire our toolkit by scanning this QR code or use the command on this slide. Here is the sample code, and here is the video of how our tool works. This is our team members of this project. Thanks.